Hi everyone, and welcome to our video on the effect of temperature on enzyme activity. So this is our fourth video in our little enzyme series, and what we're going to be doing in this particular one is having a look at that spec reference 2.1.4d, which is the effect of temperature on enzyme activity, including our reference to the temperature coefficient. Let's start off then with understanding what happens with our molecules. So we're talking enzymes still. What we actually find is that all of these different bits, these molecules, have kinetic energy. And if a chemical has kinetic energy, that means it's going to be moving. What we then find is when we heat up a substance, we're then going to be transferring more energy to those molecules. Therefore, they're going to gain kinetic energy and move faster. The faster they move, the more likely they are to have these collisions, and the more likely those collisions are to be successful. So if you go back to your GCSE chemistry knowledge, we're hitting that good old phrase, more frequent successful collisions. Now, what we then find is because we've got these more frequent successful collisions, then we've got this increased rate of formation of enzyme substrate complexes. If we've got an increased rate of formation of the enzyme substrate complexes, we're going to therefore have a faster rate of reaction overall, because the more enzyme substrate complexes we form, the more reactions those enzymes will catalyze in the same time frame. If we think back to any of these enzyme temperature graphs that we've seen over the years, then hopefully we remember that they all have this little peak on it. So when you're actually looking at your typical enzyme temperature graph, then you end up with something like this. Now, this peak is where the reaction is at its maximum rate, and the temperature at which that happens is the optimum temperature for that reaction and that enzyme. It is worth remembering at this point that not all organisms will have the same optimum temperature for their enzymes, for the simple fact that not all organisms live in the same climates. We've got a couple of some of these extreme organisms, if you like. We have our psychrophilic bacteria, which are ones that are designed to live in these really cold climates. Now, it'd be pretty terrible if their enzyme optimum temperature was around 30 degrees, because it's not getting to 30 degrees anytime soon for these guys. Therefore, their enzymes actually have an optimum which is at a very low temperature. Basically, it suits the environment in which they live. They've adapted that way. The second one, the thermophilic bacteria, are the opposite. They live at very high temperatures. Now, if we've got obviously an organism living in very high temperatures, we can't have enzymes that are going to denature. Otherwise, all those reactions that were enzyme catalyzed are going to shut down. So what actually happens here is the enzymes that we find in our thermophilic bacteria are what we call heat stable. Now that just means that they're not going to denature in those high temperatures. And the way that they have achieved that is by having this increased number of disulfide bridges, and that just gives them a more stable structure. It holds that 3D shape even when the temperatures increase. So because they've got a large number of disulfide bridges, they're heat stable, they will not denature at those high temperatures, and therefore the thermophilic bacteria can happily live in these rather toasty conditions. What we now need to understand is what happens if we increase the temperature too much for these enzymes. If we increase the temperature, what we're going to be doing is transferring a greater amount of energy to the molecule, and the more energy they've got, the greater the vibrations within it. And those vibrations can be really bad news, because what's going to happen through those increased vibrations is the weak bonds within the tertiary structure of our protein can end up breaking. If we break some of those bonds in the tertiary structure, then the 3D shape is going to start to change. And the most important site of the change of shape is the active site. So what we start to see happening as we start to increase those temperatures a little bit too much, some of those weak bonds are going to break, the shape of the enzyme's active site begins to change, and the substrate is no longer a perfect fit. Still fits, but not as well as it used to. So because of that, the rate is going to start to decrease. If we keep applying more heat to this reaction mixture, then we're going to keep altering the shape of the active site, because more and more of those bonds are going to be disrupted, and therefore eventually we get to the point that the active site is no longer complementary to the substrate. So this is an irreversible change, and that is what we mean by denaturing. So we've broken far too many of the bonds within the tertiary structure, leading to a significant change in the shape of the active site. It is worth remembering when we are talking about bonds being broken within the enzyme, what we are not referring to is breaking the peptide bonds between the amino acids. So when we talk about this process of denaturing, we are breaking the weak bonds in the tertiary structure. So these are the bonds and interactions that exist between those R groups, the side chains of the amino acids themselves. They are weak. What we're going to do is we'll apply enough energy to be able to break those, therefore the tertiary structure will be disrupted, but we do not have enough energy to break the peptide bonds. They are really strong. So because of that, we are not going to be separating amino acids from that polypeptide chain. So what we can say is the tertiary structure has been altered, but the primary structure, so the sequence of the amino acids within the protein, remains unchanged. So just go careful not to say we break all bonds, because that's a lie. We don't break the peptide bonds between the amino acids. Let's finish off then with a little bit of maths, because we know there are going to be math-based questions on the A-level papers. We'll start off with a nice easy one, or at least I think it's straightforward. To calculate the rate of our reaction, all we need to do is 1 divided by the time for the reaction to reach its endpoint. So you can get that from a table, you can get it from a graph, whatever that time is to reach the endpoint, 1 divided by that time will give us the rate of our reaction. And because this is a rate, its units are going to be second to the -1, okay, so per second basically. The calculation that we then need to do, which people sometimes struggle with, is what we refer to as the temperature coefficient. Now, this is sometimes summarised down to Q10, and what we're talking about with the temperature coefficient is the increase in rate of a process when we increase the temperature by 10 degrees C, hence Q10, because we increase the temperature by 10 degrees. Now, to work out the temperature coefficient, all we need to do is look at two rates. So we have the rate of reaction at the higher temperature, and then we divide that by the rate of reaction 10 degrees lower. So if we were comparing the rate of reaction at 50 degrees and 40 degrees, for example, what we do is the rate at 50 degrees divided by the rate at 40 degrees. So quite simply, you just read those off from the graph, and then you just plug it into your calculator. When you get that number, it tells you how much the rate will increase by. So to give you one example here, if Q10 equals 2, then that just means that for the 10 degrees centigrade increase in temperature, we are going to double the rate of reaction. If Q10 equaled 3, then that 10 degree increase in temperature would triple the rate of reaction. The trickiest thing, I think, is trying to remember which way round these rates go. People always seem to forget for some reason. So it's a 10 degree difference, the rate at the higher divided by the rate at the lower temperature. Always use a calculator, of course, because that is then going to avoid any silly mental maths errors that we make in exam situations. And then just show you're working, because even if you were to make a silly error when you're writing it down, you at least should get working marks. As always, don't forget to subscribe to the channel so you can see when more videos are uploaded, and head on over to the A-Level Biology website where you can find a range of different resources to help you in your A-Level biology studies.